introduce Professor Correia-Pinto. He is a pediatric surgeon in Portugal, chief of pediatric surgery in Braga. And I know him from his research in congenital diaphragmatic hernia and lung development, so we'll talk about that. But he's also famous for new innovations in surgical minimum invasive procedures, which we talked about yesterday, in surgical grand routes. And he's really an inventor. I got to see him as the pediatric surgery MacGyver. And you know that he's really creative. And yeah, I'm curious to hear what he has to say about his take on lung development and the future for maybe fetal therapy.
So Richard, thank you very much all of you for your presence. And I'm very glad to be here. And I hope you are here to hear our talk, and not only for the new device that you have here. And I try to deserve this opportunity. And maybe this is the, if you look at my first slide, there's no name. You already introduced that I'm speaking about congenital diaphragmatic hernia. And I have this here just to show you that how a surgeon, a pediatric surgeon, became teaching physiology, or professor of physiology became pediatric surgeon, and our disease can make this together. So my first slide, I will put this first, is just to, when we start studying a disease, that in this case is a congenital malformation, and we look for history, look for everything, we see this description from beginning of 18th century that was saying that this disease was nothing that it was seen in other children, that it could be perceived relieved by anything that we should to eat, nor by advice of any learned physician. It is interesting that when I began my residence, I feel the same. I feel that I could not do nothing for these babies. Interesting, my feeling today, and at the time was the same, is that we can save or help these babies to overcross the neonatal period, then they became quite normal. They are not, almost no sequela on this baby. So these babies deserve that we invest on research to help them to survive and to overcross the neonatal period. So what you're talking about, we are talking about diaphragmatic defect, a morphological big defect that for the surgeon is quite easy to repair. It's 2 or 3 stitches and that's all. So the problem is not a surgical problem. This is
You shouldn't say that.
So the most classical and complex disease is the left side CDA, but there are other entities that are, maybe they are related or maybe not, they are not so related, but actually they are also diaphragmatic defect, the classical Morganil array anterior defect, central defects, eventuation that is a different entity, but for sure this posterolateral defect is the most serious and severe congenital malformation, and our focus is on the lung compromise of this newborn infant. So what we know about genetics and diaphragmatic defects? We know that we have from the morphological point of view, from the anatomy point of view, we have a diaphragmatic defect with sac and without a sac. With the sac is much better for clinical than without a sac. And when we have a sac, we can be anterior, central, posterolateral. When without a sac, almost all of them are posterolateral. And now we do something that, if you look and try to find on the literature some about genes, we found that there's some correlation. It's not very strict, but we feel that there are some genes more associated to posterolateral without sac, and there are other genes more related with these other more benign forms. What is interesting is to see that if you look only on the position of the defect, it works. It is blocked with the light. Don't worry. I'm happy with this.
New technology.
So, if you look on these genes and try to correlate with the position of the defect, we also found some somatotopy. So there are some genes related with a different kind of position of the defect. So now we move forward. Interestingly, when we try to collect all these genes and look to the organs more affected, and what are the organs more affected on congenital diaphragmatic babies, are the lung and are the diaphragm, and also the heart. That's another story that I already had the opportunity to study, but I did not bring it to the slides to not become too complex. So, what I'd like to show you is that if you look on this screen, and this is coming from a colleague of Richard from Rotterdam, this slide. If you look here on this scheme, you have a perspective of the lung, looking on mesenchymal. This is a mesenchymal and epithelial. And if you look on the diaphragm, you have the muscular substratum and the muscular layer. What you see is that the same genes that I was reporting 2 or 3 slides before and speaking about the defects, they are here. And you see COOP-TF, FOG, GATA4, RAR, retinoid receptors, so they are there. So it seems that some important genes for muscular or the diaphragm development are the same important for the lung development. And here we can discuss if it's more important a muscular substratum or the muscular. Here we can discuss if it is more important the mesenchymal or the epithelial. But by now we don't have real an answer for this. So what we could resume this introduction is that the recognition of various factors that may cause the effect of diaphragm and simultaneously affect the development of other organs, namely the lung and the diaphragm and the heart, for instance, explains the range of variability of patients with CDH, regards the morphology of the defect, severity of pulmonary hypoplasia or associated malformation. So we should have several factors that could lead a disturbance, could be genetic or from the environment, and we get in a certain period of time during gestation, during embryonic development, something goes wrong that affects the lung, that affects the diaphragm, that could affect the heart, could affect other organs. But then the embryo survives. If he survives, you try to compensate, to cope with this acquired defect. And we will see when we get that. So if this embryo survives, so he will grow until the end of gestation. For our surprise, and maybe not, this fetus grows quite well. So in utero, they are okay. They grow perfect until the end of gestation. Why? Because the major defect on them are lung, and the fetus does not need the lung to grow. So the problem will occur when it starts to breathe, and on the first breath, the lung is not there. So it starts this terrible cycle that we see on NICUs, that is, we have pulmonary hypoplasia, so that is as a component alveolar also vascular. You get tissue hypoxia, distal tissue hypoxia, you get metabolic acidosis, pulmonary hypertension, both concur to this terrible finding. Right ventricular or some heart dysfunction, right and left vascular shunt, more hypoxia, more acidosis, and the babies die on this cycle. So you need to do something to revert if you are acting postnatally, or you need to do something on pulmonary hypertension before they arrive to enter on this cycle after birth. So, trying to understand what happened, why the lung does not grow, there are some interpretations, and here on the room we have Richard that gave 10 years ago, or more than 10 years ago, a huge contribution to understand this hypoplasia, the mechanism of this hypoplasia. So we could have an event, we always think of on this straightforward way. We have a primary event that could be genetic or from the environment, and then we have the defect, and then we have hypoplasia. People were thinking by this way. The surgeons were always thinking by this way. So we have mechanical compression, so we have a hypoplastic lung. What Richard introduced, and it's a nice theory or hypothesis, is that we have a primary event that creates the diaphragmatic defect, but also disturbs the beginning of the early development of the lung. So since the beginning, the lung is suffering. And maybe the heart is suffering. And then the gestation progresses, and what happened is that you have, by more or less by that place, the diaphragm should close, but it is not closing. So if it's not closing, the abdominal organs goes up on the thorax and start compressing the thoracic organs. Who are the thoracic organs? The heart and the lung mainly. The lung is very soft and suffers a lot, so became more affected, and you have a second event that promotes retardation of lung growth. The heart is more stiff and can contract, so resist much more to this compression of the abdominal organs and can recover. So the lung at the hand, it's very interesting when we look for heart studies on fetals made by obstetricians, it seems that the left heart is small. But when you look on studies on postnatal, you see that the heart is normal because it could recover. It's not suffering from the second event. And the second event are the mechanical forces introduced by the abdominal organs going up on the thorax. Okay, so this is what we have. So, as a surgeon, trying to help these babies, what we can do. After birth, my feeling is that it's too late. The neonatologists made a huge effort developing new medications for pulmonary hypertension, for ventilate better and better in a soft way these babies. But even nowadays, we have a huge mortality rate on these infants. So we need to find something during the prenatal period to help to increase the lung. What would be the best strategy? And this is not new. Actually, this is being discussed since the 90s. So people from UCSF trying to develop the fetal intervention, or fetal treatment, but it failed. But what we have is the fetal intervention could be surgical intervention, and here we could have two kinds of intervention, the diaphragmatic repair or tracheal occlusion. The diaphragmatic repair failed at all, and the tracheal occlusion is a controversial procedure. It's something very strange that the physiologist described in the 60s, and the idea is to occlude the trachea, and occluding the trachea to promote lung growth. There's a debate, or there was a debate, if the lung was just distended by liquid and pressure, or if it is really growing or not. The feeling today is that the tracheal occlusion promotes proliferation of the lung cells. I don't know if this lung is totally normal, but this is what we can offer at this moment, the only thing that we can offer to these babies. And the other possibility is to do medical treatment. And here there's not so many things to offer, even that experimentally we have these kind of studies trying to that retinoids, vitamin A administration is good for reverting or for preventing the lung hypoplasia, and could be an attractive hypothesis. The problem is that the retinoids are very teratogenic. So the window doses to work is very strict. Everybody is very afraid to use retinoids in the mom because it could be very dangerous. And there's another argument against the retinoid is that if you give the retinoids quite early, during the lung, the congenital malformation development, it's okay, you can prevent. But in humans, you are not in the fetus. You don't know if this fetus will get or not the disease. When we get the disease, it's already too late. So you are on the second trimester, you detect by ultrasound, and you find the defect. So you go there and try to treat with retinoid. The effect is not the same. The big effect of the retinoid is in the rat when you give it yearly on gestation during the beginning of the malformation. So even I'm a surgeon, I believe more on medical than on surgical treatment for CDH. So we try to follow this strategy. And all my all my experiments on this disease are based or influenced by this intriguing experiment that I performed a couple of years ago. So here what we have is in vitro lung explants culture, and then we collect from control or normal rats, from fetal rats, the lungs, as well the lungs from nitrofen pups exposed to nitrofen. And then from both groups, we extracted protein content. And then we took this protein content, or homogenate, and tried to make new cultures with normal lungs, and with adding these proteins extracted from normal fetus or proteins extracted from hypoplastic lungs. And for our surprise, or maybe not, what we got at that time, what we get is that if you add proteins to normal culture with normal lungs, the lung becomes bigger than in sham control. But if you have the protein content from hypoplastic, the lung is still bigger. So when we measured or count this, there's no doubt that if we culture explant with the proteins extracted from hypoplastic, it promotes more the lung growth than protein content extracted from normal. So this means that the hypoplastic lung has on its medium some growth factors that are trying to compensate. They know that they are hypoplastic and they have some growth factors, some substance in content that will do. So we tried, based inspired on these experiments, looking for novel physiological regulators of lung development. And it was tempted to do this kind of analysis, to put all the protein of the hypoplastic lung and in a 2D gel trying to find major difference. It's not an easy analysis, but when we perform this in rat model, immediately identifies something like this. There was a protein, identified as myosin light chain 1A, that was missing on nitrofen exposed rats. So we try to see what kind of protein, or where it's being expressed, and is expressed not in all smooth cells, is selective, so only on the arteries is expressed, not on the smooth muscle cell of airway or even the veins, it's mainly on arteries. And in explants, if you block the expression of this gene, in fact, you could block the growth of the explant. So it works like not a growth factor, but it is important for lung development for sure. I don't know if it's a growth factor. And here started my idea that angiogenes could be also very important for fetal lung development. But anyway, as a surgeon, this is very good. Gave at that moment a nice paper, but it's nothing for the baby. So we need more. So this not represent a step forward. So I try to go deep on lung development physiology and try to find other way to overcross this problem. And we look, if you go on lung biology, it's a different world with a lot of genes, with a lot of crosstalks between the epithelium, mesenchymal, endothelium, muscular layer, muscular cells. So, and at that time, I was fascinated with one of my colleagues, one of my previous PhD students, about FGF10, because at that time, a paper came out on the, so it's a major regulator or a major morphogen of the lung. And the paper came out on the American Journal of Physiology saying that on explant, if you have FGF10, you get a bigger lung. So that's what we need to do. And I convinced Alan Flake, that is our other pediatric surgeon in Philadelphia, to do this kind of experiments with us. And the idea is to overexpress FGF10 on fetal lung, trying to promote lung growth on the fetus. So we developed a very thin methodology to inject the fetal rat, the lung of the fetal rat, and here you can see how it works. I hope that I'm not doing a mistake. Yeah. So a micropipette, you see the heart, the lung, you go there and you could inject. So we have a strategy to reach the fetal lung, and now we have to build a virus, and we build a virus that could promote expression of FGF10. And when the first experiments started to come out, I was keeping in contact with the PhDs, and they started surprising me. They said, there's something wrong with these lungs, because they are, I don't know if they are bigger or not, but they have cysts. You know, I could not believe what you say with cysts. So what we're seeing in image control, like a human being that does ultrasound control of the fetus, of his fetus. So they could follow this fetus after injection, and they started to detect some cysts on the lung. You see here? See here? And then if you remove at the end of the station the lungs, you see this defective lung. And if you go to see what is happening with the virus, see, where was acting the virus, because it was associated with GFP, you see that the virus was acting on the same, place. You see? So now I will move, and then we try to do a complex and expensive experiment that was trying to inject yearly injections during gestation, late injection during gestation, central injection, and peripheral injection. And what you have here is a resume. So if you do yearly injection, proximal, central injection, you get huge cysts, unique or two, but huge. If you more distal, you start decreasing the size of the cyst, but increase the number. If you start doing late injections, so you increase the number, but you decrease the size of each cyst. So if you look, if you are familiar with the lung pathology, this resembles other entity that is not CDH. This is congenital CCAM, congenital lung congenital cystic adenomatoid malformation. Okay. Now that I know that you changed the name, so these are the pups. You see here. So when they breathe, very quiet, and this is gasping. This one. And if you take this one and to go to the MRI, you see that there was a huge cyst. So what we discovered was that we failed again finding a treatment for CDH babies, but maybe we could give a contribution for understanding better the whole spectrum of CCAM morphology. So now the idea what we propose is that maybe CCAM is based on a disturbance of FGF10 expression, and if you do a yearly injection or overboost of FGF10, you get bigger and bigger cysts, but in a reduced number. And if you do a late or peripheral injection, you get more cysts, but very more smaller. You know. But anyway, we failed again on trying to find a way to treat. So we tried to move forward, and here it was a study that took a lot of a lot of experiments with the rats, because here we tested, by opportunity, we're running other studies in the lab with renin-angiotensin-aldosterone, and suddenly we found that angiotensin was a huge growth factor, was promoting a lot of growth in fetal explant. This was found by opportunity, but if you look to this explant, this is a control explant, this is a treated explant with angiotensin, and if you are familiar with this methodology, is bigger even than the retinoid or even with FGF10 exogenous, with TGF-beta. Most of the factors cannot increase from 12 to 22 the number of peripheral buds. There's only a few growth factors that can induce this. So I look to this and try to understand better what is happening. So we dissect all the system, all the receptors, so we try to identify the angiotensinogen where it's being produced, where are their receptors, where are the enzymes, and we found that if we block, that this effect was induced by blocking 82 receptors, because there are two main receptors, and this is mimicked by PD 123319 that is produced by Pfizer, and they were very kind of it because they sent it to us because it's very expensive for experiments, and they gave us enough product to test this, and actually by using this 82 blocker you can induce explant glandular. But would it work in vivo? So the next step was to find the right dose and the right strategy to study in vivo, and with the micro-osmotic pumps instead on the mother rats, I put this 82 or this PD 123319. I'm afraid that I mistake some number, but what we found is that it promotes the lung growth in vivo too, what is very good. So you see here all the groups that we get on this experimental model. You have the control with saline, the nitrophen, I think I start to need you. Yeah. So here, well, it doesn't matter what. As a resume, if you look to the right, you get around between 10–12% of increase of lung parenchyma. What could be the difference? So 10 to 15% on the increase of lung parenchyma means that's not significant increase, but could be the difference between survival and non-survival. Because these babies with more 10 or 15 or 20% of lung parenchyma, they can survive, they can ventilate, can be operated, and then we release them on their infancy. So we became very enthusiastic with this idea, and I promoted more studies on this. And the idea was to test the real function of this lung. This lung is bigger, but is working or not. So we tried to do these survival studies on this difficult model to do that, because the CDH in rat model is very aggressive too, is very severe. So we tried to study the survival and also to collect. So we tried to have a huge number of rats to do survival studies, and it seems that the PD treated animals, they survive more. Note that we are speaking about minutes because all of them die. But the most convincing data comes from when I pick up the pups, I decapitate and take the blood from the neck, and I do some gasometric studies. And here it's evident that the PD treatment animals are much better at 5 minutes of life than those without treatment. So this is a simple experiment that convinced me that maybe we found a way to move for some clinics, because this 82 receptor has another additional advantage, is that the 82 receptor is a fetal exclusive receptor. It's not being expressed in adults except in disease. So the renin-angiotensin-aldosterone system have in the fetus express 82 receptor, but in adult, for instance, in mom, 82 receptor is not being expressed. So this could be a way to restrict the effect of our agent to our fetus. But we need to move for more clinical now, maybe in humans, to see if this is a real contribution or not. So where we are at this moment is that we try to understand better the lung development, lung growth, and this drawing try to explain what we found during this last decade in research. We found that inflammatory mediators are important. They promote growth, they promote repair, they can do repair. Namely, we studied most of these GP130 family of cytokines, and based on these findings of this study that I presented to you about PD, what we found that is that we have a crosstalk between the vessels and the airway tree. So maybe what is happening on the vessel could control much more the airway tree that we thought before. And before I close my presentation, now we have also other studies trying to define that is. This is a different issue, because when we start to do research, even I was always very focused with the idea to do some translational research, find something to promote. There's always people that fascinate you with more basic science, and here the idea is to study how the cells orient in the space and in time differentiation, and these are cycling genes that they are expressed in waves pattern. So they start expressing, then disappear, and 1 hour and a half they start being expressed again, and they, like a wave, they run all the explants from the beginning, from the central, the trachea, until the distal tip. But this is more basic. It's not the way to find the solution. Anyway, I also promote this kind of studies. And so, thank you very much. And if you visit Portugal, or in the north of Portugal, this is our hospital, and this is our medical school, you are very welcome. Thank you very much.
Thank you very much. It's always tough to use some technology at first, but you did a brilliant job, so thank you very much. So you've left ample time for discussion and questions, so let's open it up.
Okay.
Klaus?
You mentioned a whole series of genes that may have an influence, but you didn't say anything on the effect side. I presume the effect side of any particular mutation in any of these is relatively small, or if not, could you envision doing plasma fetal DNA analysis of all of these together and predict which one might be?
Yeah, that's well, very interesting question. What you are asking me is what is the cause of the disease? We don't know. My feeling, I don't know, I think that you think that the same way, we should have different ways that can concur to introduce this defect. At the end, independent of the cause, we should disturb the retinoid pathway. Because everything that we do to disturb the retinoid pathway gives CDH, could give CDH. You know? Most of these genes are somewhere linked to this pathway, but this is not the focus of my research. Saying, okay, I already accept this malformation exists, so I need to do something to repair. And to repair is not to prevent. If I was working on prevention, I would work on that. But I need to. So I accept that the malformation is there. I need to do something to repair. To repair, I can repair the diaphragmatic defect, but I need that it allows me to operate the baby.
But you would like to diagnose it early.
Yeah. So what you are saying is that you can screen the plasma of this mountain. There are several groups in Rotterdam and Leuven that they are trying to find to screen what you are saying. But I think that that is a different topic, you know. So one thing is to, maybe it could be interesting to see if, of course, the cause, but we are trying to work later. So what we can do if we have the malformation.
Andrew?
So I found that experiment with the conditioned media from the CDH lung quite fascinating. That you got this sort of paradoxical finding, what seemed to be paradoxical, that enhanced growth or promoted growth. I guess in thinking about that, to me, one of the things that the thought that that might be demonstrating is a deficiency in the receptors for the factors in the hypoplastic lung. So they can't respond because they don't know the stuff is there. Do you know, is there any evidence that the receptor for growth factor X, Y, Z, probably many,
What
maybe
we
at the
have
root of
found,
hypoplasia?
yeah, we try to understand that the finding that what we found is that sometimes, because when we have a hypoplastic lung, we try to identify some growth factors, say they are decreased, so they contribute, they are the cause of this hypoplasia. What we found is that sometimes you are trying to investigate this kind of growth factor, and they can increase on the disease.
Yeah.
You know, and this opens another discussion, more conceptual, that is something on the lung that understands that is restricted in growth, and starts a process to recover, like a compensation. Like I don't know if there are, I know that there are some neonatologists or pediatricians here, so the idea is very similar to what happens in pediatrics with the catch-up growth phenomena. I'm tempted to interpret this data by this approach. So there's something that the lung or the organism recognize that is wrong, that is too small, and try to initiate, to start a process to compensate the growth. And that's why we found some growth factors that can increase. But regarding the receptors, I must confess that I did not investigate, but it's a nice possibility to.
Okay, well, let's go there, and there, right down that road. It's a very curious road.
Yes, just to follow up on the retinal story. So, has there ever been any association studies done looking at, say, women that take topical acne medicine and association with CHD?
Yeah, not only with CDH, probably with CDH, but women that take with some creams from dermatological proposals, they have a higher incidence of some malformations, namely anorectal malformation, and that's why people is so afraid to use this kind of products in pregnancy. You know, this is a point. The other point is what we get from the literature, from very old experiments or studies showing that in moms that deliver babies with CDH, they have a decrease in retinol levels, blood retinol levels, and these kind of studies were repeated in retinol, and they found the same. So there's something wrong with the retinoid acid pathway on, for sure.
I think it's more pronounced in the babies than in the mothers.
Yeah.
Right. Yeah.
There's something wrong on retinoid, and it should be the cause. The cause should be linked to the retinoids.
I sort of have the same question, but
Were you guys talking to each other?
Just
yeah,
Osmosis, okay.
telepathy. Another question I had was, so what happens if you repair the diaphragmatic hernia and these lungs start growing again? Do they ever get to their full potential, and do you know what factors are involved in that catch-up growth that you talked about?
Yeah. You know that I will divide my answer in two parts. First, what happens when we repair the baby? So that's an incentive that is, what we observe is that the remaining hypoplastic lung grows in, if you do an X-ray 1 month, 2 months later, it's equal to the right side.
Okay, but
So it recover.
pretty.
That's no doubt about that. And if you go to some functional studies, it's not normal, but it's close to normal. And anyway, you look to the baby and the infant is quite okay. If you do not have very aggressive with ventilation, if you do not have severe infections, if you do not induce BPD, the infant will have a normal life. The other aspect is what happens, which growth factor. We don't have that answer, but I was discussing with Richard that we should do that from the experimental point of view, trying to find the leading pathways that promote the physiological growth. And you touch right on my message on this presentation. I always try to identify what the nature is doing to recover and trying to use that pathway to promote physiological growth or to burst physiological growth of the lung. but we not have data on that. I'm sorry.
Yeah, I'm a clinician from basic sciences, but you know, for us, it's not the lack of lung, okay, it's the pulmonary hypertension. So then, and that is the thing that these babies suffer from down the road, okay. Now, some of that may be induced because they think that the vent went up, okay. But some of that may be induced by that. But then, if the pulmonary hypertension is the key thing down the road, then that's probably the key thing in development. So the business of crosstalk and the vascular development has to be
yeah, that's an interesting question, but I'm always I'm tempted to interpret this data as that the pulmonary hypertension is a postnatal event. I already made, yeah.
Okay, I don't
No,
know.
because the pulmonary hypertension is physiological for the fetus.
Yeah.
You know, because the oxygen level is very low, and the pulmonary vessels are very sensitive to oxygen. When you start breathing, the content of oxygen increase, and it should relax. That's what we know from normal physiology. And what happens on these babies is that I did not present all the data, but on rats, I dissect the right ventricle, the free wall of the right ventricle of the heart of this fetus, and in utero, before birth and after birth, and on that free wall, I measure load markers, overload markers, on the free wall. And what it found is that before birth, there's no difference between control, nitrogen exposed animal. 6 hours after birth, or half an hour after birth, if you go there, so the nitrogen exposed and CDH pups have a huge increase on these load markers, whereas the remaining they don't have. So the severe pulmonary hypertension is something postnatal. The other is, there's of course prenatal hypertension, but it's physiological. You know? What we observe on these babies is that they do not relax. And the other question is why they do not relax? Because from morphological point of view, we find that there are thick muscular layer around the vessels. We don't know why. We find that. But I don't know why we have this thick layer on it, because why this baby did not decrease or became thin, the muscular vascular vessel. Should be an event, finding contributing to the hyperreactivity of the muscle, of the vascular muscle to oxygen or hypoxia.
Joe?
Something you just said kind of sparked my interest, that if you have excessive muscular arteries, I'm wondering if in this congenital defect you have an imbalance between epithelial and mesenchymal transition, and that your drug that blocks the
You should direct that question to Richard.
So I'm wondering if the drug that blocks the angiotensin AT2 receptor is somehow blocking EMT transitioning, giving you more parenchymal cells in the lung.
Yeah, it's interesting that these animals treated with this, the muscle layer is much thinner. It's not normal, but close to normal. That's yeah, you open. That's an idea
I think
to study.
angiotensin 2 would promote more mesenchymal vascular smooth muscle cell development. If you block that, maybe you'll keep more epithelial cells, which would become lung parenchyma.
We've seen in I did some recombination studies, and we saw that the fibroblasts were always the problem, and we see more mesenchymal phenotype. That's what the microRNA, so that's important.
Because myodine and GAD4 are very mesenchymal. And they're, I'd love to make an appointment.
This is the purpose of research trials, to stimulate new areas of research. Well, and I understand that there is potentially a Canada-European collaboration coming out of this, so that's good. And don't forget his invitation to Portugal. I'd also like to say we're very honored to have Dr. Susan Parsons here with us. She gave wonderful grand rounds this morning. She's director of the Health Institute and Center on Child and Family Outcomes at Tufts Medical Center. So thanks. I guess it is the time to visit Winnipeg. So great. So please come up and visit, ask more questions. There are a few apples left, a bit of Caesar salad, and thank you very much for coming and giving great.